Hi, my name is Jackie Wang, and this summer I interned at the Scott Lab studying neurogenesis and neuromigration in songbirds. And this is what I worked on this summer. Can new neurons be created? This is a pressing question, especially for diseases such as Alzheimer's and Parkinson's. In most mammals, such as us humans, neurogenesis, which is the creation of new neurons, usually occurs in early development. However, the onset of many diseases like Alzheimer's usually occurs long after birth and long after humans have lost the ability to replace their dying neurons with new ones. On the other hand, in adult zebra finches, neurogenesis and neuromigration, which is a vital step of neurogenesis, continues to occur widespread into adulthood, especially in the high vocal center, which is also known as the HVC. In vivo two-photon microscopy is an imaging method that has been historically used to study this phenomenon, but is limited to very shallow, superficial regions of the brain. And due to this constraint, many deeper regions of the HVC, which may exhibit different migration patterns, are left unreachable. Therefore, the goal of our experiment was to use this novel prism implant in the brain in order to image deeper into the brain to investigate the difference between a superficial region, which is like a shallow region, and a deeper region of the brain. And through analysis via MATLAB software, we found no statistically significant difference between four characteristics of neuron migration: speed, distance, displacement, and tortuosity. Tortuosity is like the overall windiness of the movement. And these findings suggest that imaging superficially in the zebrafinch brain is a good representation of overall neuron dynamics. We also found that imaging using this novel prism is good for neuron tracking, which may be used as a novel technique in future research to identify the cellular and molecular mechanisms of neurogenesis and neuromigration, with the long-term goal of finding a therapeutic cure for neurodegenerative diseases.